is Jason Hayes. I'm a graduate student in the CCN lab at FIU in Miami. This is a pre-recorded presentation for the 2020 meeting of the Society for Mathematical Psychology. My research explores how changes within neural population codes can be inferred from psychophysical threshold studies. Many researchers are interested in inferring changes in population codes as a result of cognitive or learning factors. For example, Sorensas and colleagues used fMRI to distinguish changes in encoder responses with attention, where attention to specific orientations affected the tuning of populations in visual cortices. Zhang and colleagues also used fMRI to distinguish changes in encoder responses that occurred with learning. Garcia and colleagues used EEG to differentiate changes in encoding channel responses due to attention over time. But there's a problem. fMRI and EEG can only measure the underlying population codes in an indirect manner. These indirect metrics have difficulty separating some of the encoding mechanisms through their population responses because they're too similar. These problems may make you wonder why most human research has been performed using solely neuroimaging. The research presented here asked a simple question. Are psychophysics a good supplement to study population codes in humans for researchers interested in how neural population codes change as a function of cognitive or learning factors? In our research, we're interested in exploring whether specific changes in neural population codes between cognitive states can be inferred from behavioral tasks with the help of standard population models, like those used in computational neuroimaging literature. While some researchers have done similar research, they tested a limited range of possible encoding changes, which we expand upon. As you can see, the models used in these simulations have several parts. The encoder consists of a set of channels, each with a tuning function that responds maximally to stimuli near the channel's preferred stimulus. The encoder's job is to create distinct representations for distinct stimulus values. The output of these channels, the population response, is noisy, as you can see by the error bars. The decoder transforms the noisy neural channel's outputs into something that can be used by the brain, the ultimately perceived stimulus. However, the decoder doesn't receive perfect inputs from the encoder's channels. In the decoder's distribution of stimulus estimates, the imprecision is represented by the width or standard deviation of the Gaussian function. According to signal detection theory, this standard deviation is equivalent to a discrimination threshold corresponding to a sensitivity or d' of 1. Thus, the standard deviation of the distribution of decoded stimuli is the same as psychophysical thresholds in our simulations. And these thresholds can change depending on the encoder channel properties. So then, what encoder channel properties did we modify? For all of our encoders, we represent channels tuning functions with Gaussian curves which have three properties: the height, width, and center. The height represents the channel's maximum responsiveness. The width, which is in standard deviations, represents the channel's coverage. The center represents the channel's preferred stimulus. The encoder uses many channels to create distinct responses for distinct stimuli. As you can see, adjusting channels' properties affects the overall response of the encoder. The baseline will consistently be represented in blue. To represent neural noise, we use the Poisson distribution, as there is evidence that neural noise is approximately Poisson, with stronger responses having higher variability. You can observe the noise difference by comparing the error bars at the two peaks. The higher peak has more variability than the lower peak. While there are other possible mechanisms of change, the literature mentions each of these types as benefiting decoding precision. Depending on how the neural codes are altered, they can worsen or improve the discriminability of stimuli differently across the stimulus domain. For example, increasing the responsiveness of channels at the target usually improves discriminability around the target and for nearby stimuli. On the other hand, decreasing the width of channels at the target may improve discriminability around the target but decrease discriminability for nearby stimuli. Because different neural changes produce different outcomes, our goal here was to determine through simulation whether the different mechanisms of change proposed in the neuroimaging literature could be distinguished using psychophysics. While previous research have distinguished between a couple of mechanisms, no one has attempted to distinguish among all of them. So let me give you a brief overview of the mechanisms that we included in our simulations. Gain can affect each channel's maximum responsiveness. If a change is nonspecific, then the change occurs uniformly across the channels. If a change is specific, then the change has greater effect near the target stimulus, which is consistently 0 degrees in our simulations. Tuning can affect each channel's width, with specific and nonspecific mechanisms applying the same way. Tuning shifts can affect each channel's preferred stimulus, moving them closer or farther from the target. Suppression reduces responsiveness of surrounding channels. Suppression plus gain is the only combination of changes we included because previous literature suggests that it may be a mechanism of change that occurs through visual attention. In this study, we used an encoding-decoding observer model to simulate how a number of mechanisms of encoding change would affect the decoding imprecision, as measured through sensitivity thresholds. We created a baseline homogeneous population encoding model which had tuning functions with a width of 12 degrees, a height of 10 Hz, and positions evenly spread along a circular stimulus domain within -90 and 90 degrees. We simulated both a sparse population with 10 channels and a dense population with 20 channels. Neural channel noise was simulated using independent Poisson noise, a common choice in the previous literature. Statistically optimal decoding was implemented via maximum likelihood estimation, which produces a normal distribution of stimulus estimates centered at the true stimulus value with a standard deviation proportional to the sensitivity threshold. As you can see from this example, the variability of the baseline distribution of estimates is wider than the variability of the distribution that came from the encoding model that was affected by specific gain. The bottom row has a more precise decoder near 0 degrees. We measured imprecision, again equivalent to thresholds, along 2 dimensions. The target orientation consisted of 181 stimulus positions from -90 to 90. The level of external noise consisted of 375 levels of external noise from 0 to 0.5. For each mechanism of encoding change, we also tested 3 magnitudes of change with both a low and high density population. For specific mechanisms, we also had 3 different effect fall-offs: 1/3 width, 2/3 width, and full width, where the full width effect faded by 90 and -90 degrees, the most distal values from 0 degrees. There were 50,000 iterations per threshold. As expected, the simulation suggests that neural codes affect the psychophysical thresholds across the stimulus domain. By comparing how the baseline thresholds change relative to each manipulation effect, you can distinguish each manipulation. As an example, you can observe the difference in the manipulation effects between specific gain and specific suppression with gain. The thresholds for specific gain are always at or better than baseline. The thresholds for specific suppression with gain are worse than baseline at the edges of the domain. Here we have each manipulation alongside the baseline. Almost every orange line is different from the baseline in a different way. However, nonspecific gain and nonspecific tuning are not distinct from each other if you only look at thresholds along the stimulus domain. Furthermore, specific gain and tuning are not distinct in every case, although they are in the majority of the cases. Even still, you can separate the other types by how they differ from baseline, and you can still tell that the type falls into one of these subgroups, meaning you can narrow it down to either specific gain or tuning or nonspecific gain or tuning. Specific suppression plus gain was an interpolation between specific suppression and specific gain. If the gain or suppression mechanisms are imbalanced, then it's too difficult to differentiate it from the others. Thankfully, running additional psychophysical experiments can help us distinguish the two problematic types. The stimuli in this figure exemplify different levels of external noise. Depending on how the neural populations are changed, we can expect them to respond differently to different levels of noise. In other words, you can determine how the threshold of discrimination at a given stimulus, for example an orientation of 0, increases as static external noise is increased. Considering nonspecific gain and nonspecific tuning, both types improve performance at low levels of noise. However, as the external noise increases in nonspecific gain, thresholds become more similar to the baselines. As the external noise increases in nonspecific tuning, thresholds become worse than the baselines. While the threshold versus noise curve can clarify which nonspecific changes are occurring, the function is represented by a cumulative curve, and unlike the stimulus curves, there are a limited number of ways in which the curves can differ. Threshold versus noise functions still have problems differentiating specific gain, tuning, and suppression plus gain from each other, and also have trouble separating specific suppression from inward shift. In conclusion, psychophysical studies can help distinguish changes in neural codes. Most of the models can be differentiated via psychophysical thresholds, but keep in mind that the assumptions must be made to make inferences about population codes from indirect measures, just as in computational neuroimaging. Using behavioral paradigms, participants' thresholds can be compared to the model's thresholds to infer what changes occurred in the population codes underlying human performance. Multiple experiments may be required depending on which coding changes will be considered among alternative hypotheses. Like neuroimaging, psychophysics makes assumptions and is an indirect measure of the neural codes, but can increase our ability to separate possible mechanisms of encoding change. Using psychophysics, we can gather additional data to reach better conclusions about questions that ask about what is changing in neural codes involved in behavior. I'd like to thank my mentor Fabian Soto and my lab mates. Thank you all for your attention.